Welcome to today's episode where we examine the interplay between chromatin architecture and gene regulation. Our guest, Dr. Ryan Corces from the Gladstone Institutes, investigates how 3D genome organization and epigenetic landscapes shape cellular identity and contribute to disease. By combining single-cell epigenomics, chromatin profiling, and computational modeling, his work reveals how dynamic regulatory elements orchestrate transcriptional programs in development and cancer. Join us as we dive into the mechanisms that link genome structure to function at single-cell resolution.
Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
So today I'm happy to welcome Ryan Corces from the Gladstone Institutes on the show. So welcome, Ryan.
Thank you.
So to ease into the topic, a question I like to ask every guest to start off the podcast is, how did you become interested in biology in the first place and then pursuing a career in science?
Yeah, I guess I have a story that is somewhat common in the sciences. Both of my parents were actually scientists. So I grew up in a family where both of my parents were running labs. My mother was a faculty member at the University of Maryland in Baltimore County. My father was a faculty member at Johns Hopkins. And so, dinner table conversation would be about science and struggles with the lab and funding and things like that. One might think that would push one away from a scientific career. And my parents actually often encouraged me to pursue other things. I had a kind of burgeoning interest in being a computer engineer, computer scientist when I was, going through high school, and they kind of pushed me down that path and pushed me away from science in many ways. And I was a stubborn kid, so that was probably the last thing that one should do. So I ended up kind of gravitating back towards biology at every turn. And so I did some internships both on computational things, but also in the lab when I was in high school. And that's really where I got my start as a scientist, was both in their labs and then in other labs, through internships.
Yeah. So for those unfamiliar, could you introduce yourself and your current role?
Yeah, so I am an assistant investigator at the Gladstone Institutes, which is a small nonprofit research institute that's academically affiliated with UCSF. So I have a co-appointment in the neurology department at UCSF. And I'm an assistant professor there. And my lab we started up around 2020, kind of in the beginning of the pandemic. And we've been researching the non-coding genome, and specifically a lot about genetic variants in the non-coding genome and how those contribute to diseases, specifically Alzheimer's and Parkinson's disease.
So we will get into that in a minute. But from what I could read from your website, even though you just shared with us that you have a science background, your path into science was maybe not that linear, but maybe I'm on second thinking it's maybe different. But maybe you can share that, because after your graduation, you spent a year living with family in Spain and teaching science to bilingual elementary school students, and before you then started your PhD. So I guess this was not your family when I hear the background now. So how can you talk about the step of your career and what motivated you to do this?
Yeah, so I had done my undergraduate training at Princeton. I had been working in the lab, for a couple of years at that point. I had known that I wanted to go on to do a PhD. And so I had applied to PhD programs at the end of my undergraduate career, as if I was going to enter into the PhD program immediately after graduation. But then I got this idea in my head that this was one of the last times that I would ever be able to take time off and do something that wasn't science. And it was something that really appealed to me. My father was born and raised in Spain. And so he actually did all of his training through graduate school in Spain and then came to the United States for his postdoc and never left. But all of his family lives in Spain. And, we, of course, were well acquainted with our family and we would see them every now and then. But there's a pretty big ocean in between the East Coast where I grew up and Spain. So, it wasn't enough really for me. And I really wanted to connect with that side of my family's culture. And in truth, also feel more fluent in the language because it wasn't something that was very comfortable for me at that time in my life. And so I started looking around for what could one do if one took a year off and went to a foreign country and how would that all work? And I found what they called a language and cultural exchange program that was run by the government of Spain, where they would bring in native English speakers to work as teachers or teaching assistants in the bilingual school system there. And teaching has always been something that I loved. It's actually one of the reasons why I decided to pursue this career, and having the opportunity to both live in Spain and live in the city with my family, my father's family, and then also be able to teach kids and connect as part of like an educational system is something that really appealed to me. And so I applied to this program and was accepted, and actually ended up going to Spain, and my brother also participated in the same program. So we went together, and it was an amazing experience, in part, as a 22-year-old, I only had to work, 20 hours a week or something. And so I had a lot of time to explore both Spain and Europe and other surrounding areas, Turkey, Egypt, did a lot of traveling and kind of personal growth in that time, but also, being forced to learn how to connect with a student or an audience that is at a completely different life stage than you, right? Like, I was teaching kids ages 4 through 8. And so not only are 4-year-olds extremely different from 8-year-olds, but they don't think about problems in the same way that you do. And so being forced to come up with lesson plans and connect with the students on that intellectual level, I think has really driven me as a mentor and a teacher towards, a very different approach to those things. So it was an amazing experience. And I was very grateful that Stanford allowed me to defer my admission for a year and do that. And I often do encourage students to consider taking that time if they can. These days, it's a little bit harder, at least in the US, because I think a lot more is expected of people when they start graduate school these days. But
yeah, this would have been my follow-up question, right? Did this influence now the way you run your lab?
I think it has. I mean, it's hard to know where all of the influences come from. I often actually credit my PhD mentor with a lot of my own mentorship style, but I think every bit kind of contributes in a way. And so, it's not like I'm treating my lab members like they're 3-year-olds or 4-year-olds or something in elementary school, but I do think that in the way that I prepare material for talks and try to connect with an audience when I'm talking to them in a seminar, it does have flavors of thinking deeply about the material that you're trying to convey. So I do think that it has impacted how I run my lab and how I, also present the work that we're doing.
So when we transition into your PhD work, there you provided the first genetic and cellular proof that AML, or leukemia, evolves from sequential acquisition of mutations in one or a hematopoietic stem cell. So can you then talk about how you got started and what you found doing those experiments?
Yeah. So this, I guess, dates me a little bit, but when I was starting my PhD, it was 2009, and the first big papers of sequencing a genome, not the Human Genome Project, but taking someone's genome and sequencing it on a sequencer, came out a year before that, and so genomics and the actual field of especially cancer genomics, where we had a very good feeling that these mutations were the important things driving this disease, and if we could just sequence people's cancer and we would be able to figure out what the drivers were. That whole field was coming to life when I started my PhD. And, in some ways I did just kind of happen to fall into that, but being armed with a kind of classical computer science training and a molecular biology degree, I felt like this was the ideal place to deploy those two skill sets. And so I started in a lab with the intention of doing cancer genomics work and trying to understand this kind of mutational evolution of cancer. And, it started out as a purely kind of genetics genomics project where we were sequencing and digging through that sequence again to find drivers of cancer. And what we were looking at was this very early stage of leukemogenesis, where we have this hypothesis that the hematopoietic stem cells were the key cell type where mutations could be acquiring over many years. And the rationale for that was that stem cells are long-lived, and if you're going to have multiple mutational events, they're probably going to occur in a long-lived cell, or they're going to confer a long-lived nature to a shorter-term cell. And what we ended up finding was that these mutations that were occurring at these very early parts of leukemia development were all occurring, almost all, in modifiers of the epigenome. So, different proteins that control the epigenome in some way. The cohesin complex was a key one, but DNMT3A, so DNA methyltransferases, and TET2, other components of this sort of epigenetic machinery, they were all being mutated early on in this disease pathogenesis. And this struck me as very interesting, very unexpected, I guess. And so that's really what launched my interest and kind of career into studying the epigenome, epigenetics, and the non-coding genome, was this kind of fundamental discovery of these early drivers of leukemogenesis and acute myeloid leukemia specifically.
So there is this theory, the two-hit theory, right? That you have to have like maybe two mutations and two different players. How did this integrate or compare to your work?
Yeah, no, it's a great question. The two-hit hypothesis, I think, really worked well with what we found, because we would find leukemia patients where they have these early events mutating these epigenetic regulators. And then the later events were more mutations that would confer a proliferative advantage. And so it seemed to have this kind of two-hit mechanism where, and this is completely conjecture, but I think others would probably agree with the theory, that the epigenetic kind of modification that was happening early on was priming the cells to be able to accept this really, really highly proliferative mutation later on. And so it definitely fit very well with that two-hit hypothesis.
Mm-hmm. Okay. You then joined the lab of Howard Cheng, and there you also met, I guess, Jason Buenrostro. Jason has been on this podcast already in episode 53. And together you worked on applying ATAC-seq, which I guess Jason pioneered to understand normal hematopoietic differentiation and also leukemic transformation. So how did this transition to your postdoc work? What were you thinking, or what was your experience, and what did you find using ATAC-seq in those areas?
Yeah, it's a very astute observation. So, the timeline of this, I guess, is a little bit fuzzy in my head because it all happened so quickly. But Jason and I met towards the end of my PhD. He had already published the original ATAC-seq method. And, he was looking for systems in which he could deploy the method and find some new biology leveraging ATAC-seq. And at the time, ATAC-seq had a lot of strengths, but one of the things that was very challenging was that primary human cell types were a little bit more difficult to get good data from than some other cell lines and things like that. And so I think he had been probably collaborating with a couple of people trying to see what would work out. He had a lot of really outstanding kind of diversity of work during that time, applying ATAC-seq to different systems. And at the time, I had just kind of come across this whole epigenetic modifier thing, and I was really interested in both how these epigenetic modifiers would change transcriptional state, but also how they would change the epigenome. And so I had been working on a project where I was sorting, using fluorescence activated cell sorting to isolate out these kind of leukemia precursor cells and their normal human blood counterparts, and really trying to understand what were these epigenetic modifier mutations doing to the pre-leukemic, as we call them, stem cells. And Jason and I linked up on that project, and also on this project that I thought at the time was really cool, and we never ended up pursuing it, but on mitotic bookmarking and some other things that we thought we could do with ATAC-seq. And he said, well, hey, why don't you just sort some of these blood cells and we'll try to get ATAC-seq to work on them. And it just worked, immediately. And so that was kind of one of the first times where deploying ATAC-seq on a new cell type just worked without any optimizations or challenges. And so we ran with it. And I kind of put the leukemia part to the side, and we started building an atlas of hematopoietic lineage data, and seeing what we could tease out of that. And in that collaboration, Jason really did the lion's share of the analysis, and I was working with a technician that Jason had to generate all of this data. And it was just like this amazing turning this crank, and every day I was sorting cells and generating libraries, and it all came together really quickly. And so it was around that same time that I started to think about where I would want to do my postdoc. And it seemed to become a very natural progression to stay at Stanford, change labs, work with Howard Chang, and kind of build out more of this epigenetic interest that was kind of growing in what I was doing in the lab.
So it's also always very interesting because you see those papers written, all right? You see the original ATAC-seq paper by Jason, you see the OmniATAC paper that you then probably wrote up because of all your experience that you did in this time, right? And then getting to know the story behind all these cooperations, that's very, very interesting to see. So was the OmniATAC-seq protocol that you published later, I guess it was in 19, if I'm not mistaken, was this based on all of this work? And how did you improve the original ATAC-seq protocol?
Yeah, the OmniATAC story is a really wild story. Jason and I had been working together for a long time at that point, and we had tried ATAC-seq on a whole lot of different cell types. And I had gotten, as I mentioned, was very interested in epigenetic modifiers in cancer. And so my first postdoc project was related to understanding the cohesin complex and how these mutations in the cohesin complex were changing the epigenome. And I had generated a whole series of cancer cell line knockouts and all these things. And I was getting really poor ATAC-seq data quality in those cell lines. And it drove me nuts because, I could grow millions of them. They were healthy, and I just could not get good data from those cells. And, over the years, we had come up with some optimizations, and we had some other observations that we just couldn't quite explain. And one of the cell lines that I was working with was K562 cells. Everyone works them, and they just didn't give very good ATAC-seq data. It was passable, but it wasn't great. And I felt like it wasn't going to be good enough for what I wanted to do. And the key observation actually just kind of came from this chance experience that when Jason was working on the single cell ATAC-seq protocol using these small chips, it was all on the Fluidigm platform. The K562 cells would actually give really high data quality. And so in bulk, they gave poor data quality. In the single cell assay, they gave good data quality. And the only real difference is that on the Fluidigm chips, PBS was being added into the mixture. And so that ended up being one of the keys, for whatever reason, to getting good data quality out of some of these harder cell types. There was a few other optimizations that we kind of deployed, but I ended up basically testing in isolation and then in combination, every single modification that we could think of to this protocol. And I did it all, more or less in a week. I tested a whole bunch of things, a couple hundred libraries, and threw them on a MySeq and got a few reads from the each library. And I remember I got this result right before I left for my honeymoon, and it would have been in like October or something like that. And it was clear as day. Like, one combination of things was just leagues better than anything else we had ever tested. And so I went on my honeymoon, and I came back, and then I prepared a couple hundred more ATAC-seq libraries to kind of flush out the story. And I presented it at lab meeting, and I remember Howard saying, like, "Wow, this is great, but like, what do you want to do with this?" And I was like, "Well, I want to publish it." He's like, "Well, you've only changed like one buffer and a few detergents. Like, it's not that impressive." I was like, "Yeah, well, the data quality difference is huge." And so we wrote up a short 2-figure paper to Nature Methods. And, from October, when I got the first result, the paper was accepted in June or something really, really quick. About 6 or 7 months, given that I had taken a couple of weeks off for my honeymoon. So the whole story came together really quickly, and it really just is kind of exactly what you said. This like accumulated wisdom over many, many hundreds of ATAC-seq libraries. And, now that protocol, it's been really widely used. I think it's helped a lot of people, and it's just a few buffers that have changed, but it really did make a big difference for the data quality.
Yeah. Kits are based on the protocol. So,
yeah.
So you said that, and I might jump ahead because I've written down the question, but you somehow switched gears during your postdoc, right? So you did all this ATAC-seq work, but you then moved on to pursue other epigenetic questions, right? So maybe we can leave it a little bit of an open question and let you do the talking.
Yeah. So, I guess there were a couple of things that kind of happened contemporaneously. So one of the reasons why I started, after the OmniATAC paper was published, Howard Chang and Will Greenleaf, they started this collaboration with the Cancer Genome Atlas, and they pitched trying to get high quality ATAC-seq data from primary frozen tissue samples. And that was kind of all happening around the same time that I was doing the OmniATAC stuff. And because I had been doing all this protocol optimization, they said, "Hey, why don't you take on this project and try to get this to work in these tissue samples?" And, I got a good amount of help from some other people in the department, trying to figure out, how do we isolate nuclei from these tissues? How do we, actually work on some of these frozen archival tissues? But once we figured out how to get the nuclei out in a very clean and high throughput way, the ATAC-seq part was pretty easy. And so that was kind of one of the bigger stories that we put together, was this kind of many hundreds of cancer samples and trying to understand, what could we get out of the ATAC-seq data from those tissues, especially because, I think ATAC-seq on its own is a little bit of an amorphous genomic readout. It doesn't tell you too many specific things, but it tells you a lot of other stuff, and it's really powerful when you combine it with other data types. And on the Cancer Genome Atlas side, they had RNA-seq and methylation and all these other things from the same samples. And so it was a really unique opportunity to start combining these modalities in primary human tissue. And it was also an amazing opportunity to work within a consortium where, these people are professionals. They know everything they've been working on these samples for, a decade, and they know everything, and they can. It was a really collaborative and great opportunity to work within that sort of environment. So that was kind of all still continuing in my cancer, phase, I guess, of my scientific training. But and I don't know if this is what you were hinting at, but I also did then kind of transition to studying neurological disease, which was a pretty big change in what I was focused on. And that's if
I can interrupt quickly, because before we move on, I have another follow-up question. So if you have all those information, right? You have ATAC, you have RNA-seq, you have DNA methylation. You get like obviously the 3D structural information of chromatin, right? But you also maybe can extract like a time information, right, of subsequent happenings, what is happening on a transcriptional site, right? You know, if is it methylated, is it transcribed, is it open? So I mean, this opens a whole lot of more information, right?
Yeah, it definitely does. And I think, one of the things that I guess on the methylation side, it's always been a little bit tricky with ATAC-seq because they're usually quite inversely correlated. More methylation, less accessible. That sort of pattern. Certainly on the transcriptional side, it gave us a lot of power to start connecting these regulatory elements to the genes that they were correlated with, right? So we could look for correlations across hundreds of samples and ask, when this regulatory element is active, what gene expression is changing across these different tumors. I think the other thing that was really interesting was the fact that, in cancers, there's lots of copy number alteration. And so we could start to understand how those copy number alterations impacted the chromatin accessibility landscapes and how that correlated with the RNA-seq landscapes. And there was a lot of really interesting things that could be done with combining those data types.
Now we can come back to the transition that you did, because this then also plays into you setting up your own lab, right? At the Gladstone Institutes.
That's right. So, again, all of this was happening around the same time, but one of my very good friends and lab mates, Ansu Satpathy, who's now at faculty at Stanford, we were sitting back to back in Howard Chang's lab, and we were always talking and running ideas by each other. And he was a resident in the pathology department, and he was approached by the chair of his department to try to get ATAC-seq to work on brain tissue. And because I was doing all of this optimization for tissues anyways, he roped me into generating some of this data. And it was just a few more libraries amongst a sea of hundreds of libraries. So I said, sure, why not? And so we made the data. It looked great. And then he said, I was like, here, you can have it and you can present it to the chair. Like, I don't need to be involved. It's like, no, you should come to the meeting and meet the guy and show him the data and tell him about it. And so I went to that meeting, and it's really a meeting that changed the rest of my career. The chair of pathology is Tom Montine. He's still the chair now at Stanford. And he was thrilled. He was excited about applying these technologies to brain tissues. And I said, look, Tom, I'm a cancer biologist. Like I'm laser focused. I want to be a faculty. I need to keep going down this path. And he said, well, what if I hire a research associate to work with you and you train them and they generate the data, and, it's not so much of a investment for you to make. And I said, okay, sure, we can do that. So, one thing led to another. The data looked really interesting. And also Tom was just a phenomenal mentor. We would oftentimes meet on the weekends, and he would walk me through things and teach me things about the brain. And, he really just kind of helped launch this whole side of my career, in particular with helping me write a K99 R00 fellowship on deploying some of these epigenomic technologies in Alzheimer's disease tissue samples. And that's what really kind of launched me into this career path. It's just this kind of random happenstance interaction with a lab mate, and then someone who was a really fantastic mentor that invested in me and kind of saw the potential that I would have to contribute to that field.
So I guess the opportunities are always there. You just need to be ready to take them.
Yeah, that's right.
So can you maybe talk about the roles of those mentors? Because I guess it's not just like being there on site in the lab, doing the work, helping on site, but also like being there on the weekends, as you just said, walking maybe around the lake, talking about things. How did that, or how important is that?
Yeah, I think different mentors give you different things for sure. and I think as a trainee, the most important thing is to make sure that you have mentors that will give you each of those things. You can get it all from one person, but it's maybe a little bit more rare. And so having kind of a team of people that's mentoring you is important. It is hard to find people that will invest in you unless you're also like investing in them. So it is a two-way street. But, for example, my postdoctoral mentor, Howard Chang, I've never met anyone who is as good as Howard at this kind of vision of both the current and future science. He has this knack of knowing what's coming and being able to help you contextualize the story in a way that everyone sees its impact. And so, for that side of my scientific whatever know-how, I learned immensely from that environment. And then on the other hand, Tom Montine, he was much more of kind of a boots on the ground type of mentor, where on a week to week basis, he was teaching me things and making connections with other people in the department to fill in gaps in the story and things like that. And my PhD mentor was I certainly model my own mentorship style after him, but he really knew when to let me run free and when to rein me back in, and like how to encourage my curiosity, but in a direction that would benefit me. And so I think all of these mentors give you slightly different things, and it's just important as a trainee to try to recruit as much of the good that you can out of the people that are around you.
So you then set up your own lab focusing on like Alzheimer's disease, I guess. And the next paper I found was published a few months ago in May 2025, describing choir. I guess it's this pronounced. I don't want to describe this myself because I had trouble understanding it from the abstract, to be honest. But maybe you can talk about this computational tool for single-cell analysis.
Yeah. So as a lab, we, in the very early days of the lab, got a couple of different funding mechanisms to perform really massive scale single cell data generation, both in Alzheimer's and in Parkinson's disease. And as we were generating this data, a lot of which was just still unpublished, but will hopefully get submitted soon. One of the key challenges that we felt like was still unaddressed in the field was that when you have all this single cell data, the ultimate goal is to identify groups of cells that, define either cell states or cell types. And most of the tools that are currently out there suffer from either this problem of over clustering, which you have a single cell state or cell group that's actually chopped up into a bunch of sub states that aren't real. They're just driven by noise. Or under clustering, where you have, multiple cell types or cell states being considered as one. And that balance between over and under clustering is often caused by a really heavy reliance on user-defined parameters. And so the single cell field just got very comfortable with this idea that when you were going to perform clustering and identify cell types and cell states, that you were basically going to have to use biological intuition to drive the decisions of, what cell groupings you were going to call. And, that's fine. It works. It's very laborious, but it's also just not very reproducible. And so we set out to develop a computational tool that would take a lot of the guesswork out of defining cell groups and cell states. There's a lot of other kind of contemporaneous methods that were proposed to do some of this. One of the big ones is what people refer to as reference-based mapping, where you have like big atlas scale data sets of the whole human body, and then you can label your cells in your data set based off of annotations from some other data set. And those do work, maybe they work less well in the context of disease, where cell states might not be as well mapped to the normal human condition. But what we wanted to employ was more of a statistical framework where you could say with statistical certainty, whatever that means, that this cell group is biologically distinct from this cell group, and that those distinctions wouldn't be dependent on how many cells were in the cluster or cell group or things like that. And a very talented PhD student, Katrine Sands, took on this project. Really, it was her own brainchild. She came up with the idea and drove it all the way to completion and produced a software tool that I think really does help and can be deployed for some of these atlas scale, many millions of cell data sets now. And so certainly within our own lab, we use it quite a bit and gotten a lot of great feedback from other people in the field.
So when you study either Alzheimer's or Parkinson's, it's rarely studied apart, right? You always find people working on both of those diseases together. Why is that the case?
Yeah, I don't know. It's an interesting observation. I think because they are quite similar in terms of the human condition, right? It's loss of cells in the brain, and we don't really understand why, and they have different phenotypic outputs, but they probably have somewhat related drivers. But I think the other thing is that those two diseases exist on a spectrum, right? So there are people who have, pure Parkinson's disease and others that have pure Alzheimer's disease. But then there's a lot of people that exist in between. Most patients exist in between, where they have Alzheimer's disease, but they also have pathology that is, reminiscent of or consistent with Parkinson's disease, or they have Parkinson's disease and they also have pretty severe cognitive decline. And so, I think the attractive part of studying both diseases is that not only do you study those two endpoints of the disease, but you can also try to understand what's going on in this kind of murky middle ground where people have mixed pathologies, and really those mixed pathologies are the vast majority of patients that come into the clinic.
Okay, I see. So imagine you are due to submitting a grant proposal tomorrow. What would you have written into that?
I am due to submit a grant proposal in 2 weeks, so I can maybe talk a little bit about that. So one of the things that we've been very interested in the lab is how non-coding genetic variants contribute to disease risk. And, I got into this by studying the results from genome-wide association studies, which you profile the genomes of tens or hundreds of thousands of cases and tens or hundreds of thousands of controls, and you look for genetic variants that are statistically associated with being in the cases column or the controls column. And those studies almost universally identify common variants because you need to have enough observations of a variant in order to statistically assess it. So they have to be, more or less present in more than 1 to 5% of your patient population. And cumulatively, at least in Alzheimer's and Parkinson's disease, those common variants explain a relatively small fraction of the overall genetic predisposition to disease. And, then that leaves this question, that this kind of phrase that people use a lot, which is this missing heritability. What is the portion of genetic risk that we haven't been able to explain yet? And so in the case of Alzheimer's disease, we think of Alzheimer's disease as being about 70 to 80% heritable. Meaning that genetic risk is driving a very large portion of your likelihood to develop the disease. But we can only explain about, 1/5 of that genetic risk based on common variants. And so the rest of that piece of this pie is explained by something else. It could be structural variants because we haven't really been able to tackle structural variants that well in disease yet. And it could be rare variants, things that are present in less than 1% of the population. And so the the grant that we're hoping to get funded is proposing to study the role of rare variants in disease. But the challenge is that when we say rare variants, uh, each person has about 10,000 rare variants in their genome. And, 98% of those are going to be non-coding. So we're really talking about rare non-coding variants. And we like to contextualize this problem as, not only is this haystack massive, not only does everyone have 10,000 of these variants, and we can't just like use normal statistical association because there's too many of them and they're not observed frequently enough. So not only is this haystack massive, but the needles that are hidden within that haystack are hard to find because they're in the non-coding genome. And so through collaboration, especially with Anshul Kundaji at Stanford University, we've been using machine learning models to train models to predict non-coding variant effects. And then, with that as our kind of prioritization scheme, we can go through millions of variants in a few hours and start to address the, haystack is massive problem using machine learning based prioritization and prediction of which non-coding variants are going to have effects. And that has been really powerful in our, preliminary analyses. We can actually kind of whittle down that haystack to a point where there's many fewer variants, and that really increases our statistical power to run association tests on rare variants. And it also opens up the potential of studying smaller familial pedigrees where they have high penetrance disease, but they don't have a known genetic modifier. And so, if you use segregation analysis, you might end up with 1,000 variants that segregate within this family. But if you then use your machine learning tools, you can get that down to maybe 10 or 20 variants. And now you can really start to study those in the lab. So what we're looking to do in the future is these kind of really massive scale prioritization efforts with machine learning, and then following that up with functional genomics in the lab to understand how all of these genetic alterations are impacting the non-coding genome and gene regulation and cellular state in the context of disease.
So for the last 43 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
Oh, interesting. No, I think we've covered a lot of the things that have driven me to this stage. I guess, the thing that I think gets lost when we think about publications is really the role that people have played along the way. From people who have gave me the opportunity to work as a high school student. So Jonathan Eisen, who was at the Institute for Genomic Research many, many, many years ago, to my undergraduate PI, Colleen Murphy, to all of the people along the way that have helped me become a scientist. I think, the thing that I think gets lost in the publications is the people that were there along the way. So we talked about Jason, another hugely impactful person in my scientific career. I think that those people are really the story, and I'm glad that we had a chance to kind of highlight some of them along the way.
To finish, what is your personal definition of epigenetics?
Yeah, that's always a tough one because I know deep down that ATAC-seq is not epigenetics, but I want it to be included in that umbrella. For me, epigenetics is everything that controls cellular state. And so in the lab, we think about epigenetics as basically everything that isn't the actual changes to the DNA sequence. So that's a pretty broad definition, but it at least allows us to include ATAC-seq as an epigenetic method.
Because ATAC-seq wouldn't picture the heritability of the marks, or?
Yeah, it's not DNA methylation. It's not a histone modification. So it's not something that's being, added onto the genome. It's not epigenetic. It's more of a readout of what proteins are bound there, and it's, so I know deep down that true epigeneticists think of ATAC-seq as not being purely epigenetic, but
Right.
I like to include it in that umbrella.
Well, we like as well. So thank you, Ryan, for your time and for being on the show.
Yeah, my pleasure. Thank you for having me.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.